Welcome to the fourth Russell and Robert Moody lecture in the series on the birth, life, and death of the brain. I'm happy to see as many of you here as there are, given all of the weather and other problems that we've had in recent days and weeks. We are very fortunate to have a neuroscientist who studies the brain of children from age 6 through adolescence. I'm sorry about the adults, we can't do anything for you. It's too late. I've had a chance to meet with Silvia today. She's a delightful conversationalist and also has many interesting ideas about our brains and how our brains work and how our minds work. Silvia Bunge is an associate professor of psychology, has her own lab at the University of California, Berkeley, where she does neuroscience research, brain imaging. She had a rough time finding a college to go to, so she went to Stanford. And then first she went to Yale, Stanford, then she went to Yale. So she's covered both coasts. She has won numerous awards. She's a member of distinguished research organizations. She's published many articles, edited several books. I'm not going to waste your time running through her CV, but let me tell you, it's one of the most impressive documents I've seen in a long time. And we're all looking forward to hearing what she has to say about children's brains.
Okay, good evening. Can you all hear me in the back? Okay. If you're here for the shark movie, I'm sorry, it's the wrong thing. I really do appreciate you coming out here tonight, and I also want to give a special shout out to Bill Winslade, who invited me here tonight. So we're going to talk about brain development. All right. Why do or should we study the developing human brain? Well, the first reason is out of curiosity. How is it that we become our unique, complex, fascinating selves? Second one is, it's cool. There's been an explosion of research in this field, and there's been a rapid pace of discovery using cutting-edge technology. The third is that we provide critical information for a lot of different people who have to make decisions about kids or who work with kids, including parents, childcare providers, educators, clinicians, policymakers, judges, and of course, children themselves. And all of us need to be informed consumers of the onslaught of media and advertising, 90% of which is incorrect. Right? Okay. So a very brief overview of your brain before we dive into tonight's topic. Your brain, as you probably know, has two hemispheres, two sides. Each one has 4 different main segments called lobes. Tonight we're going to focus on the frontal lobe and the parietal lobe because they're really important for higher cognitive functions of the kinds that I study, like reasoning, decision-making, and more generally goal-directed behavior, which is our ability to stay focused on a task and get it done. If we take a slice through the brain like this, we can see the intricate structures inside. In particular, we can see this gray outer area here that's hugging along the perimeter. That small area is called the gray matter, or the cerebral cortex. And all the white stuff inside is called white matter. Let's take a closer look at the gray matter of the brain. And what you can see is that it's filled with neurons, filled with nerve cells, as well as other kinds of cells. And these are the main type of cell that you're going to find in the cerebral cortex. And if we look even more closely, we can see a particular neuron, and it's got several main parts, subdivisions. The first of these is the dendrites here, and these are receiving chemical signals from many, thousands of other neurons. And then the cell body, which carries out basic functions of any cell, but also where all these different signals are integrated to determine whether or not this neuron is going to fire, is going to send a signal. If it does so, then it sends an electrical impulse very rapidly down what's called the axon here, all the way down until the terminals here, where it then sends chemical signals out to other neurons. Okay? And these neurons are very tightly packed into the cortex. This is a cubic millimeter right here. That's what that represents. There are about 50,000 neurons in that area alone. And over the entire brain, there are about 86 billion. Okay? So it's a very complex structure. If we look more closely at the white matter, if we peel away the cortex or the gray matter, you can see this rich set of fibers or tracts that are running in multiple directions here. And if we look more closely at that, what we see is that these tracts are made up of many, many thousands of axons. And those axons themselves are nerve fibers, right? These axons surrounded by myelin, which is a fatty tissue, a fatty sheath that serves as an insulator. So just as cables are insulated, the wires that you see, in order to protect electrical signals from decaying, these too are insulated. Okay, so today we're going to ask 4 main questions. How do we study the human brain? How does the human brain develop? How does brain development support cognitive development? And what role does a child's environment play in brain development? Okay, so how do we study the developing brain in humans without actually opening up the skull, right? Until about 20, 25 years ago, it really was not possible almost at all. Magnetic resonance imaging, or MRI, has been a huge boon to the field of developmental cognitive neuroscience. And so you probably are familiar with this machinery. You can lie down on a scanner bed and get any part of your body imaged that you need to, including your brain. And you can lie down in there, and you can lie down in there and you can see, you can view images, you can press buttons while you're lying down in there. And so MRI can be used to measure brain structure. For example, right here, we've got Homer Simpson's brain. You can look at its anatomy by taking static images. And you can create, these gorgeous 3D renderings based on multiple static images of the brain. And then we can also study brain function. So the different parts of the brain that do different types of things, like that represent donuts, for example. Okay? And how do we do this? Well, we take advantage of the fact that the brain, when different parts of the brain are active, that blood is flowing to those different parts of the brain. So you can essentially take a movie where you're looking at the blood flowing to one part of the brain after another as people are performing some kind of task. And in that way, based on how you've designed your task, you can figure out what a particular brain region is doing. Okay. So MRI is being used now to study brain development, and this is an image of a newborn's brain. And by comparison, this is an image of an adult's brain. This is not Angelina Jolie's actual brain, okay? Her publicist told me I had to say that. But what I think you'll notice that's pretty striking is how similar the newborn brain is to the adult brain, right? It's smaller, but all the basic parts are already there by the time you're born. So we'll talk about what actually is changing afterwards. In some ways, actually, the brain is developing very early. As a toddler, we already have 60% of the adult brain volume. We have almost as many neurons as we will ever have. It's downhill from there, folks. And we have more connections between neurons, or synapses, than an adult has. So just to show you here, these are those cortical neurons that I mentioned: newborn, 1 month, 9 months, 2 years, and adult. And you can see that at 2 years, it's an even denser, intricate web of connections than it is in adulthood. And then by age 9, we have reached the same brain volume as an adult. But if you look under the hood, there's still a lot changing after that, supporting cognitive development. Just a whirlwind tour of the way that I see cognitive development is that it proceeds from more simple to more complex skills. So at the basic level as a newborn, you're learning to perceive the world with your various senses and to react on that basis, to react.